So good morning, good afternoon, everyone. We're back in what we're reading today in the Mycorrhizal South America Mycorrhizal Network and also the IMS. Before starting, we have a great guest. There's a interesting interview today. I want to say that if you are interested in interviewing someone that you have read an interesting paper, please contact Cesar. I will send an email that you can contact, and please do it. It's a very interesting experience, and then you meet people and do a nice interaction with them. But today we have a very special occasion because this is the first time we interview the same person twice. So this is because she's doing a excellent job, very interesting papers, and today we're gonna go with one of them. So first, I will introduce Camille Delavaux or Delavaux. I don't know what's.
Yeah, you got it correct the first time. Yeah.
Good. And we're talking about the papers. See, with a big team of people have published in communication biology called Mycorrhizal Feedbacks Influence Global Forest Structure and Diversity. And yeah, first of all, thanks for coming, Camille. And yes, let us know what is this paper about.
Yeah, well, thanks for reaching out and thinking it's worthy to interview about. So yeah, this paper, I don't know if I want to share it now, but is broadly testing for if there's a systematic difference between mycorrhizal types. So we only considered AM plant species and EM plant species because there was recurring evidence at smaller scales, like regional scales or continental scales, that EM plant species experience weaker negative CDD, which I can try to explain maybe, because before this so there's this concept that basically being surrounded by conspecifics can reduce recruitment, growth, and survival of species. And so, yeah, it's basically the reason we thought there might be a difference between the mycorrhizal types is because EM might be more specific in their host fungal relationships, which might actually counteract pathogen-driven negative density dependence. So basically, pathogens might repel conspecifics away from their adult or like older individuals because they want to escape their pathogens, but actually mutualists could pull them closer and kind of aggregate instead of repel. But we thought this might be strongest in EM plant species because they might protect directly against pathogens and are more host specific. So that was the broad setup here.
No, that's very interesting. Maybe you can share it, and you have a very nice figure that displays these two very interesting concepts that maybe you can.
Yeah,
this one.
This one?
Yeah.
Yeah, so this conspecific repelling or density dependence has mostly been studied in terms of pathogens. So that's what we're depicting here. So there's this like high density of pathogens that then basically species want to escape that cloud of host-specific pathogens and then go further away. But then, there's this general kind of emerging concept that mutualists, especially mycorrhizal fungi, might drive plant species to actually clump and kind of counteract this pathogen-driven density dependence. And people, tend to study, I may be going off, tend to like just look at density dependence and attribute it to pathogens without explicitly testing it sometimes. And so, the net effect is the like joint action of pathogens repelling and mutualists probably bringing species closer together. So,
Cool. So this was the main aim, the main idea, and how have you tested it and what you have found?
Yeah, so I guess I don't know if I'm scrolling too much, but we used this really detailed, amazing forest inventory data from across 43 forest geosites. So they measure the DBH, identify the species, and geolocate every individual in the plot above 1 centimeter in DBH. So it's insane. So we only used one census here, but we divided each species into size classes. So that's our adults and saplings. So it would be great, yeah, we talked about this in the discussion, to have actual time, 2 time points and explore, recruitment through that, through time. But here to include more locations, especially tropical locations that cannot, don't have multiple censuses. We use the spatial approach. But so we basically modeled, it might be easier here to think about what we actually did, but we modeled sapling density as a result of how many conspecific adults, or the larger size class, is in a given quadrat, which is 20 by 20 meters within the big plot. And we did that for every single species in every single plot. I don't know, I think that's thousands, I forget the exact number. But yeah, overall, so we first figured out this change in per capita sapling density with increasing adult density, and then eventually tested whether it was different between the 2 mycorrhizal types. So that's what we're doing in the C here. Mm-hmm. So across all these species everywhere, including, we included site as a random effect, and also I can show you a figure by site, and it's really consistently EM plant species experience a lower negative CDD. So they're less repelled by their own species, if you will. Do you want me to share the other figure, or is it not helpful? I don't know.
Oh, well, no, I was wondering, maybe just explain it in the figure, the green is the EM, right? And the purple is the AM. So when the density increases, then there is the EM will have higher density, per capita seedling density, right? Which means that what will
well, so you'll notice they're both the mean predictions are still under 0, so there is still this pervasive density dependence in most plant species, but basically EMs suffer less from it. So if you look at A here or B, they're kind of just on different scales. A just declines, the purple line, which is AM, just crashes faster, so they are do worse under their own species faster, as you add more and more adults, whereas EM are less harshly affected. So the slopes are always negative, but I mean, not every single case, but generally that's a consistent trend. But just EM are less affected by this density dependence, basically.
That's very cool. That's very cool result. Yeah, this consistency. Yeah, and you explain this result, and I think you add to. I mean, there are 2 main reasons for that, right? The difference between AM and EM in this in this CCD, and is related to the mycorrhizal role, right, on ecto.
So yeah, here we're just looking at tree mycorrhizal status, so what type it's presumed to be, right, based on the best checklist we have, but we're not explicitly looking testing for mechanisms. But the reason we hypothesized that there would be this kind of consistent difference is because EM fungi are thought to be relatively more host specific or have a greater affinity, so they might differentially benefit the conspecific species relative to heterospecifics, which is necessary for the kind of reverse negative density dependence, the weakening of it. And they also are thought to protect against pathogens, although we know that AM also do that. So yeah. So it's just kind of a broad scale test of this consistency, but there definitely needs to be future work to actually see, is it due to greater host specificity in EM or,
Well, this makes sense, right? Because it's what we know in general. Is it like general pattern that we assume they are true, that they need to be tested, and maybe it changes in different contexts. And I was wondering whether you have thought about the specificity, because you talk about the specificity to the mycorrhizal fungi, and then you can also have some specificity for the pathogens, right? Which is the other leg of the idea. Have you considered that, or do you think this will change also the framework or the pattern?
Well, there's no way to tree classify pathogen groups, so that will require actual, looking at the microbial community, I think. But it is, commonly thought that higher host specificity in pathogens leads to stronger negative density dependence, whereas here we're proposing that higher EM specificity leads to like counteracting that negative density dependence. But
that's very cool. So it will be very difficult to do the pathogen specificity, as we will have also a community in its tree, and so it's
and to actually test specificity, you would have to compare the one conspecific compared to every other heterospecific and show that it has a higher relative abundance and or effect on that species. So, but it could be done in a kind of like experimental field situation, maybe where you consider a smaller amount of species. But
what about this is very clear pattern, very very interesting result, very logical explanation. Do you think you can do something similar with mycorrhizal, not with rhizobia bacterial symbiosis? Like this will also could include it here.
Yeah. And I guess, when you first mentioned it, it did sound interesting, but then how would you?
Yeah,
it would just be like.
Also, I'm thinking also that it doesn't really fit into the hypothesis, because the rhizobia could also be AM or non-host, or not sure there is any ecto, so it will fit into the AM, and I'm not sure how it will change this mycorrhizal anti-mediate. So maybe I cut this question, no worries, because I'm not sure.
Now I'm like, because the two categories would be non-n-fixing and n-fixing, like associated.
It would have complexity.
So maybe things that are associate with a beneficial symbiont that can accumulate would experience a weaker negative density dependence than things that don't. But I don't know if that's maybe. We ignore non-mycorrhizal here, so that might be interesting to compare also. Because is it lower than AM? Like, is AM helping somewhat to counteract, or?
Did you
maybe?
Maybe you don't remember. Just as you said that you ignore the non-mycorrhizal. Do you recall if you find many non-mycorrhizal trees? Maybe there is, for sure there is some, like the Proteaceae, but I'm not sure there is many.
I am very sure it's under 10%, but that's still pretty high. But it's in our methods, we like explicitly talk about what percentage is removed for certain reasons. Like also if we don't have enough replication, like for every species, we required 10 points to like make the curve. So if they had less, they're super rare, then they did get removed, but that's a pretty low amount. But we were mostly able to overcome that because we included every single species in one model and just had lots of random intercepts and slopes. But
so Yeah, you mentioned also that yeah, you use the best. That it has been a huge debate about which is the best data that we have and so on. And do you think this might change in the future, like the what we know about types of trees could change, and this may change a little bit the results? Or you think that you have so many trees that in either size that 10% of changes in the identity, mycorrhizal identity, will not really change the patterns?
Well, we did not actually, yeah, do like any sensitivity analysis, but in every other study, like global study that I've done that has mycorrhizal type, you can change it like 20% and the results are typically pretty robust, but that doesn't mean it wouldn't be true here. But I do think it's very like there are 2 or 3 other like regional scale studies that find the same pattern, and in every site we find the like this weakening. So I feel like it's a consistent thing, but we'll have to see.
Cool,
it would be really good to not, in general, not have to extrapolate so much, like at the family level, or for instance, like actually know what is associating with what mycorrhizal type and also where would be the ideal, like, because it might be different, even in the same species.
There are another level that I think we don't have data for that, but it would be interesting to keep in mind that we know very little about the intraspecific variation of the mycorrhizal association, and this could this could be. I mean, it probably is who knows, but maybe what we know is very robust for at this scale, but it's something that we should be open to take it into account. So what do you think is the main message of this paper?
Yeah, well, I think basically the giant message, and like for people who are not obsessed with mycorrhizal fungi, is they are probably important in mediating these, the CDD, the spore structuring plant communities and determining diversity. And so, yeah, we should try to incorporate them like all the way from experiments to global analyses to how we interpret results, and not just always go to pathogens. So, yeah.
Yeah, that's great. One thing that I like a lot of this paper and this research and your result as well is that for many decades we were so focused on negative interactions, like competition, like this pathogen, and now this and other research shows that, okay, there is mutualism, there is facilitation, and this can be as important in principle as the other one. So that's really cool. So what other research are you doing right now that you can tell us to see what will be coming?
Yeah, so when you asked this, I wasn't sure if you wanted only mycorrhizal things. But
not necessarily.
Well, yeah. I don't know. There are many things I'm working on. But so there's like the proposal that you're involved in, that's to kind of see how mycorrhizal matching might alter invasion success in the world. So like does the dominant mycorrhizal type dictate what kinds of plants are successful at invading, which basically why we care is like to try to kind of subset the types of plants that are a risk to certain places. Yeah, and I'm also doing this ericoid project that recently we got a grant for from ETH, which is really exciting. Which is the ultimate goal is to kind of compare the relative importance of ericoid fungal diversity and plant diversity in predicting plant productivity. Because usually studies just ignore the microbiome or just do plus minus microbes or like, but we know there's probably complementarity within, ericoid and mycorrhizal species, for example. And yeah, so currently we're just doing this like initial mycorrhizal growth response assay with individual plants in all of our isolates. We have 8 different fungi to just like get to know our species, but eventually we'll do a mesocosm experiment.
That's very cool.
Yeah,
that's very cool. Would you? Did I get it right that you will have a session in the ICOM about this ericoid thing, or not? Not really, or maybe
I hope so. Well,
okay,
we submitted it. Yeah.
Okay. Cool.
So yeah, most also because we're like both. So I'm working with this really closely with Laura Van Galen, who you interviewed, I think, recently or like in the last yearish. And yeah, so I'm more of an AM person, she's more of an EM person, and we're kind of just like pretending to know about ericoid fungi. But we've been talking a lot with Martin Bonnick and others like in Zurich and other countries to learn. But yeah, we thought it'd be really nice to bring everyone together and have a space to really learn more.
That's very cool. We are actually in Spain created a mycorrhizal group, and we're gonna focus on exploring the ericoid. We're gonna start with ericoid because this is the less studied and less understood in Spain. And in Spain we have like alpine species and also Mediterranean species. So because Ericacea has this bipolar thing, and we're not sure. Yeah, it's actually something that I've been interested in for many years, and we don't know actually whether all these Ericacea are ericoid. And reading this Martin review paper seems a little bit more complicated than I thought it at the beginning, but it's not too complicated. So it's
Well, yeah, I mostly have ignored them until I read this paper about like ericoid diversity shifts with nitrogen deposition across Europe in heathlands and bogland in what do they call them? Heathlands and peatlands. And yeah, I just kind of am wondering what does that mean for like biomass production and carbon storage, which is really important. Yeah, contribution of those systems. But yeah. There are other things, but I don't know. Maybe that's good.
Yeah, that's good that you keep some
top secret.
So thanks a lot for your time, Camille. That was great to talk to you. It's really nice paper, and I hope people will be more up to reading it. It's not, I have to say, it's not too long. It's very well written, and it's very, very, very interesting. So thanks again for your time, and see you soon, hopefully in ICOM.